Whole month staining of Drosophila tissues, such as testis samples, is often problematic due to limited antibody penetration. Our fixation protocol, using NP40 and heptane, permits a uniform and reproducible labeling. By maintaining tissue shape while permitting deep antibody penetration, this procedure facilitates the reproducible and quantifiable acquisition of immunostaining signals in 3 dimensions, both singly and in colocalization studies.
For diploid germ cell evaluation, 0 to 15 hours post-eclosion, anesthetize the flies by brief exposure to carbon dioxide and transfer the flies to a fly pad. Place the pad under a dissecting microscope at a 10x magnification and use forceps to remove the heads from 5 to 10 male flies. Transfer the decapitated fly bodies to 100 microliters of Ringer's buffer on a silicone-coated glass slide on a black background support, and use the 40x magnification and a pair of number 5 forceps to grasp a fly between the thorax and the abdomen. Then pull the abdomen away from the thorax and rapidly isolate the testes and adjacent tissues from the fly carcass. Once all of the testes have been collected, clean the samples of any remaining tissue and use a 200 microliter pipette tip to remove the excess buffer from around the dissected testes. Then place a drop of 4% paraformaldehyde onto the testes.